Hi class, so we're going to start out this video lecture today with a review of the lac operon, which we discussed Monday. And so I want to go over the diploid analyses that Jacques Monod performed with his colleagues. The purpose of this experiment was to look at different types of lac operon mutants and decide whether or not beta-galactosidase activity is turned on in the presence of an inducer, which is lactose, or if it is remaining off. And so on here we have our first example, which is a wild type cell. So this wild type cell is obviously going to have normal regulation of the lac operon. So in the absence of lactose, we're going to have the repressor sitting on the operator region blocking transcription. And then in the presence of this inducer, lactose, we will see isomerization of lactose into allolactose, which will bind the lac repressor, causing it to come off the operon. So this is normal regulation of beta-galactosidase with and without the presence of lactose. So if we look at this mutant here, the second mutant, we're showing you that there's a mutation in the LAC-I gene. So the LAC-I gene is what is actually going to be transcribed and translated into the lac repressor. So if we have no lac repressor, we are going to see constitutive activity of the lac operon regardless of whether or not we have an inducer present. So notice that we have our, without the inducer, we have beta-galactosidase activity, and with it, we do as well. So this lac operon is then not sensitive to the presence of lactose. The third example that I'm showing you here is the constitutive mutant, and so that's designated by OC. You can think of OC as being the same thing as O negative. It's just indicating that there's a change in the operator sequence, so we cannot get binding of our lac repressor. This is why the second and third examples here on the screen are going to have the identical phenotype of being able to transcribe beta-galactose regardless of whether lactose is present or not. The presence of a mutant repressor results in no repressor binding. The presence of a mutated operator sequence also results in no repressor binding. In the last two examples, we're showing these mutants with the combination of a plasmid. That's what the F-lac is referring to. The plasmid is introduced to the cells, and in both cases, this plasmid, we're saying, has a functional LAC-I sequence, a functional operator sequence, but the LAC-Z gene, which encodes beta-galactosidase, is mutated. So within this plasmid that we're introducing, we're trying to replace both the repressor and operator sequences as being normal. So in the fourth line on the screen, we're showing you that the mutant doesn't have a functional lac repressor, but through the addition of a plasmid that does, that protein product from the plasmid can diffuse into the cell and bind the operator sequence on this particular mutant. That's going to restore the normal beta-galactosidase activity in response to lactose. Now, in our last example, what I'm saying is we have a constitutive activation of beta-galactosidase because we don't have a functional operator sequence. Now, if we introduce a plasmid that has a functional operator sequence, we can get repressor binding of that operator sequence on the plasmid. But as I've indicated here, our plasmid does not contain any functional genes of the lac operon. So we would not expect to see a restoration of normal regulation of beta-galactosidase activity. So this is why we don't see any change in this mutant. So this is an example of being able to fix a mutation that acts in trans. So our trans-acting repressor can be replaced with another functional repressor, but the cis-acting sequence of the operator cannot be replaced with a plasmid introduction. So nothing is going to change regarding the regulation of the lac operon in this case. So to review, our cyclic AMP is going to be high in the absence of glucose. This is going to help us positively regulate the lac operon, because we know we can actually only get low levels of transcription of beta-galactosidase when we are removing the repressor, because we have to have actual positive regulation turned on to get high levels of expression. So our glucose, when present, inhibits adenylyl cyclase. That's going to reduce the levels of cyclic AMP in the cell. And so we have this inverse relationship between glucose and cyclic AMP, and this is how the lac operon machinery is going to actually detect glucose levels. So in the case of low glucose, we do not inhibit adenylyl cyclase. We're going to have high levels of cyclic AMP in the cell. That cyclic AMP can bind CAP, and remember your book refers to this as CRP, but we're going to refer to it as the CAP protein. And so upon binding to cyclic AMP, CAP's conformational change then allows it to bind DNA. And it's going to bind the DNA at what we call either the CAP binding site or the activator binding site. And that's going to be just adjacent to the lac operon promoter region. Okay. So this is also an example of how we regulate gene expression through feedback loops. And so we're receiving signals from the cell, and that's going to affect the ability of transcription factors to control the transcription of the lac operon. So in the case of the repressor, we are going to have an effector molecule, which is allolactose, binding the repressor and removing it from the DNA. In the case of the activator, we're going to require an effector molecule, which is cyclic AMP, to bind CAP. That's going to allow CAP to bind the DNA. So we have to both remove negative regulation and activate our positive regulation to achieve high levels of expression of the lac operon. And so let's go through some of these examples. High glucose is going to result in the inactivation of adenylyl cyclase. We'll have low levels of cyclic AMP in the cell. CAP will not be bound to cyclic AMP, and therefore it's not going to have any affinity for the DNA. And then we won't be able to get high levels of transcription of the lac operon. In this case, if lactose was present, we would see that low basal level of transcription. If lactose was not present, the operon would be completely off. And now if we have low glucose, we have active adenylyl cyclase that's synthesizing cyclic AMP. Cyclic AMP then binds CAP, and if we have lactose present, we will get very frequent levels of transcription. Again, if lactose is not present, this would still be completely off. So we always say that the repressor or the lac repressor wins in that situation. So in order to achieve high levels of transcription of the lac operon, you're going to need both removal of the repressor, so turning negative regulation off, and then adding in CAP as your activator to turn positive regulation on. These again are summarized here. Notice how we have basically infrequent or really off transcription as long as that repressor is bound to the operator when lactose is gone or low. That's going to result in no transcription. We can get our basal level of transcription in the middle part of the screen when we have both sugars present, because we're removing the negative regulation, but we have no positive regulation. So when we have no regulators in place, we will get our infrequent transcription or basal level transcription. Lastly, when we have low levels of glucose and we have lactose present, that is going to turn off our negative regulation while actually turning on our positive regulation. Okay, we're going to kind of reiterate these 4 scenarios. When we have high glucose, our cyclic AMP is low, lactose is absent. That's going to mean that we have our repressor binding at the promoter region, blocking RNA polymerase from moving forward, and we have no actual positive regulation, completely off. Second scenario, we have no lactose and no glucose. That means that we have our negative regulation on and we have our positive regulation on. So if we have both of those, the repressor wins and the lac operon is still completely off. Now, for our situation where we have basal expression, that's going to be the example where we have high levels of glucose, so we don't actually need to be synthesizing the machinery to break down lactose. But since lactose is present, the repressor is not on the operator. This is going to allow RNA polymerase to associate and actually undergo low levels of transcription of the lac operon. So this is our basal expression. And then lastly, that's when we have our positive regulation on, because we have no glucose. Cyclic AMP is bound to CAP, which is then bound at the activator binding site. And we have no negative regulation because lactose is present in the cell. So that's going to result in our high levels of gene expression. And so I encourage you to draw out on a table these different sugar conditions, whether or not you would expect to see beta-galactosidase expressed, and what level of expression you would see. Because in two scenarios, it will be no expression whatsoever. In one scenario, we will have basal expression, and lastly, we will have another scenario with high expression. And think about this in terms of whether or not you are turning on or off your positive and negative regulation. Okay, so we're going to move on and talk about another operon. This is for breaking down arabinose, which is another sugar that E. coli will metabolize. Now, just like the lac operon, we're not going to want to turn on the arabinose operon unless we've depleted our glucose supply. So this is another sugar that's not as easy to metabolize, and therefore we're going to metabolize every last molecule of glucose before moving on to metabolize arabinose. So we're also going to need to have regulation that modulates this operon in terms of the presence of arabinose and the absence of glucose. So we're going to have, again, see CAP and cyclic AMP be an important factor in regulating this operon. And so we see here that this is the operon, and it has some similarities and some differences to the lac operon. At the very end, we see that there are structural genes. Those are considered the genes that actually break down arabinose. And in the case of the lac operon, the structural genes were LAC-Z, Y, and A. And so we have a couple regions here where we have an operator, which is within the promoter region, and we also have an inducer site. So that's the CAP binding site, and then we have our operator region. So the interesting thing about the regulator protein in this case is that it will bind the DNA regardless of whether arabinose is active or available in the cell. But it's going to bind different locations. And so in one scenario, it will actually bend the DNA, blocking transcription of the arabinose metabolism genes. And we will see it bind in another site to help induce the expression of these structural genes. And so we expect to see that same combination of sugars result in levels that were basically off, and then we will have basal expression, which is in the third position, and then lastly we'll have very high levels of expression of the arabinose operon. And again, this table is showing what the cell should be doing, not necessarily what it is doing. And so in the case where we have arabinose available and glucose available, in theory, we don't actually need those structural genes for arabinose metabolism to be synthesized. But that, as we'll see momentarily, is going to result in basal expression of this operon rather than being completely off. And so Ara-C is going to be synthesized similarly to the lac repressor. It is constitutively expressed, so it's always expressed, and it is going to be expressed by a separate promoter region outside of the arabinose operon. And so Ara-C is going to be able to bind arabinose, and when arabinose is present, that's going to indicate to the cell that it should be transcribing those structural genes. However, we're not going to get high levels of expression of those structural genes unless we have low levels of glucose as well. So this is what it looks like in the presence of arabinose. We're going to have repression of the entire operon. So we see that there's a binding site within Ara-C that when it's not filled in with arabinose, this repressor is interacting with two parts of the DNA, and that's going to block the association of RNA polymerase. And so we're preventing transcription of the arabinose operon by DNA looping. So in the presence of arabinose, that sugar can bind the Ara-C protein. That's going to change the shape of Ara-C such that it can no longer bind that particular repressor binding site, or AKA operator, on this region of the promoter. So we have no DNA bending, and we're actually interacting with the site that is adjacent to the promoter. So it's basically like an activator binding site. So Ara-C can act as both a repressor by interacting with repressing sites to turn off the operon by DNA bending. And in the presence of arabinose, it can bind the DNA in a separate region, and that's going to enhance transcription of the arabinose structural genes. However, again, this is only going to happen at high levels when we are turning on our positive regulation through the presence of arabinose, and then we're going to be turning on positive regulation in the form of cyclic AMP and CAP. So we're removing the repressor function of arabinose, making it now an activator, and that's going to allow for basal expression. Then we'll get higher levels of expression when we include the fact that we have no glucose available. So cyclic AMP and CAP also bind at this operon promoter region to induce high levels of expression. Okay, so this is what it looks like. A couple examples here. We have the Ara-C protein that's functioning as a repressor because there's no arabinose present. And you can notice that it's interacting with the site that actually is basically blocking where CAP would bind in the first place. And it's referring to it as CRP here. Now we see that Ara-C bound to arabinose is binding a separate region on the promoter. It's now making it possible for RNA polymerase to associate, and it's going to help stimulate RNA polymerase transcription through the interaction between the CTD tail of RNA polymerase and the CAP cyclic AMP complex. Okay. Again, if we look at what's going on in the operon under these different conditions, when we have glucose and no arabinose, our operon is looped, and so that is our negative regulation is on, and we have no CAP cyclic AMP binding, so we have no positive regulation either. Now, if we have neither sugar, we still have our negative regulation on in the form of Ara-C, and our CAP cyclic AMP is available, but because the operon is looped through Ara-C, we're not going to see any transcription. Now, once we've linearized our operon through the presence of arabinose in the cell, Ara-C undergoes a conformational change. It unloops the DNA. It opens up the binding site for cyclic AMP bound to CAP. But if we have glucose available, in this case when we have both sugars, we're not actually going to turn on that positive regulation. So lastly, when we have only arabinose and we've run out of glucose, we've turned on our positive regulation in the form of CAP cyclic AMP, and we've turned off the negative regulation of Ara-C looping that operon. So please notice the similarities in the different sugars and the regulation of this operon, and compare that with the lac operon and how that is regulated. Okay, so we're going to go through our first quicker question. The way I'm going to do this is read the question, take a brief pause. That's going to allow you enough time to pause the video, work on the question yourself, and then resume it, and we'll go over the results. So for this first question, I want to ask you which of the following conditions is going to result in fully activated transcription for the arabinose operon? So what sugar combination is going to trigger high levels of expression? All right, so hopefully you got the right answer here, which is D. So under low glucose conditions, we're going to have activation or positive regulation through the action of cyclic AMP binding CAP, and then having CAP bind this operator region. And we're going to have high arabinose, which triggers the conformational change in Ara-C, allowing it to bind a slightly different site on the entire operon, and we were going to get activation of transcription at high levels. Okay, so we're going to move on and talk about an entirely separate operon. This is going to have very different style of regulation than lactose and arabinose operons, because the tryptophan operon is actually controlling the biosynthesis of tryptophan. Now, this would need to be regulated differently because what we want to do is turn this operon on when tryptophan levels are low in the cell. So it's regulated in part by a repressor. So this repressor, when it is bound to tryptophans, when tryptophan is available to the cell, it's going to bind an operator, shown in the second portion of this slide. So these structural genes here, TRP, EDC, etc., we're not going to go over any of their functions in detail, but just know that as a collective group, they are functioning to synthesize this amino acid, tryptophan. And so, again, if you have an operon that controls biosynthetic enzymes, you're going to want to synthesize those in the absence of tryptophan here. And so we're going to want to turn it off when we have tryptophan available. And so the tryptophan repressor binding DNA when bound to tryptophan itself as its allosteric effector, it actually doesn't do the greatest job repressing. It tends to transiently come off the DNA, and so we call that a leaky repressor. So when we have tryptophan present, we would expect the tryptophan operon to be completely off, but we do see expression of tryptophan due to the repressor coming off. So the cell has actually evolved an incredibly interesting way as basically a backup method to continue to turn off transcription of the structural genes, and that's to compensate for the leakiness or the inability of the tryptophan repressor to do its actual job. And so what we're looking at here in the middle of that sequence is something called the leader sequence. So this is a sequence that's between the repressor binding site, aka operator of this operon, and so it's in between that and the structural genes. So we have a couple scenarios that can go on here. Now, if we have tryptophan present in the cell, we hope that the tryptophan repressor binds the DNA, and it turns out that we will get a transcription of some portion of this operon regardless of whether we have tryptophan or not. You'll see what I'm calling an attenuated RNA here on the screen. So this is going to occur when we actually do have tryptophan available, but our repressor hasn't done the greatest job. We have a second scenario when we truly have run out of tryptophan, we have no repressor binding, and we're going to synthesize an mRNA that contains the leader sequence as well as those structural genes. So there's going to be some important parts of this attenuated mRNA or the leader sequence that gets transcribed, and depending on how that mRNA folds into certain secondary structures, that's going to control whether or not RNA polymerase continues to synthesize the structural genes of the tryptophan operon. So RNA polymerase can always synthesize at least a short piece of RNA from this operon, and then only under true low tryptophan conditions will we continue to synthesize all the way to the end to make that polycistronic mRNA encoding the biosynthetic enzymes for making tryptophan. So what we refer to this as attenuation. So attenuation is stopping something that could potentially continue. So we're stopping it early. So the sequence here has 4 separate domains. So don't think of this in terms of protein domains. These are just regions of this mRNA. Now, these different domains have complementarity to each other in that domains 2 and 3 can actually base pair with one another, and domains 3 and 4 can base pair with one another as well. And so remembering closely back to our row-independent transcription termination mechanism in prokaryotes, in that case, these complementary RNA sequences automatically folded up and bound to one another as that RNA exited the RNA exit channel. This is going to, if they have complementary sequence, they will fold as soon as they can, because in this double-stranded conformation, the RNA is more stable. So once we've transcribed domains 1, 2, and then finally domain 3 exits the RNA polymerase, it will pair with domain 2. Okay, that's going to happen under our low tryptophan levels. In our high tryptophan levels, our ribosome, which is dictated on this slide, it actually is going to pause at a region that kind of overlaps between domains 1 and 2. When that occurs, as domain 2, 3, and 4 have exited the RNA polymerase, we cannot have pairing of domains 2 and 3. Instead, domains 3 and 4 will pair under this condition. Now, the interesting thing about domain 4 is that immediately following it, we have a string of U's in that RNA. So domains 3 and 4 forming that secondary structure, followed by a string of U's, is going to result in attenuation or early stopping of transcription. And it turns out that when domains 3 and 4 are paired and we get this transcription stop signal to the RNA polymerase, it is stopping and coming off the DNA right before it was about to synthesize those structural genes. And so this is an example of translation regulating transcription of a gene. And so what we're going to look closely at is the location of the ribosome within domains 1 and 2, and how that is going to control the folding of this RNA, and whether or not we're going to get a attenuation or terminator signal for RNA polymerase to stop transcription. So if we zoom in and look very closely at this leader sequence, I want to point out that we have, it's not shown here, but we have a start codon in the beginning of the leader sequence. And soon after that start codon, we're going to see 2 adjacent tryptophan codons. So 2 codons that are going to encode for the amino acid tryptophan. Next, the other feature I want to point out is that we have a stop codon right at the end of the sequence that corresponding to the first domain. And so when a ribosome is stopping at that stop codon, it's actually preventing regions 2 and 3 from pairing. And so the tryptophan levels in the cell are going to actually control where the ribosome moves on this RNA and how far it gets on the RNA when it's attempting to translate it. And so there's 2 possibilities. One is that the RNA polymer, or the ribosome, right, starts at the start codon, proceeds to translate a very short peptide, and then if it reaches these 2 adjacent tryptophan codons, it will typically pause, and depending on how low the concentration of tryptophan is in the cell, it could take quite a long time for a tryptophan-specific tRNA to be made and make its way to the ribosome to bind here, to be incorporated into that peptide. So low tryptophan is going to make it difficult for a ribosome to translate through the adjacent tryptophan codons. So it, in effect, is going to pause there at a region that's fairly early on in that domain 1. So when the ribosome pauses there, there's enough time for parts 2 and 3 of that leader RNA to be transcribed, and they will pair with one another given that chance. Now, the other scenario is if we have sufficient levels of tryptophan in the cell, our ribosome is able to go directly through the synthesis of that domain 1, making a slightly longer peptide, and it's going to stop at the stop codon, undergo its normal translation termination mechanism. So when the ribosome is stopped at the stop codon, think of it as that these are very large complexes, and so a ribosome sitting on that stop codon is actually physically preventing region 2 from pairing with region 3. Part of the ribosome is essentially overlapping onto region 2, so that when region 3 is synthesized, it does not pair with region 2, and when region 4 is synthesized, Parts 3 and 4 immediately pair up with one another. We have a poly-U sequence, so that is a string of U's on the RNA. That would be temporarily base pairing with a string of A's on the DNA. And due to the formation of this termination signal in the form of parts 3 and 4 pairing, that's going to bump up against our RNA polymerase at a time when it has a very weak interaction through the DNA, through these AU base pairs. So that's going to cause our RNA polymerase to actually stop transcription at that point, and then it's not going to actually synthesize those structural genes. So let's take a couple other looks at this. Notice that when tryptophan is present, we are able to incorporate tryptophan-specific tRNAs into this small little peptide that's being generated. And by stopping at that stop codon, we are partially overlapping with region 2. So the ribosome is kind of covering it up a little bit, making it impossible for regions 2 and 3 to pair. So since the other possibility is that regions 3 and 4 pair to one another, when this occurs, we're going to actually see dissociation of our RNA polymerase based on the fact that we've formed a transcription termination signal. And so that's a stem loop formed by regions 3 and 4 paired together, followed by that string of U's. So this is an example of row-independent transcription termination, and it's going to be controlled precisely by where the ribosome sits on our leader sequence RNA. So remember that the leader RNA is always transcribed, but we don't actually necessarily synthesize the part of the operon that encodes the structural genes. So the leader sequence is made, and then depending on whether or not we actually do have low tryptophan levels, we can control whether or not the RNA polymerase continues to transcribe those structural genes. So when we have tryptophan absent, we are going to have our ribosome start translation and then stop at those adjacent tryptophan codons. It doesn't really matter if it pauses just ever so slightly, like it takes a couple seconds for tryptophan tRNA to show up, or if there's virtually no tryptophan in the cell and you never move forward past that little region. Either way, so low tryptophan levels causing any sort of pausing of the ribosome is going to allow regions 2 and 3 to be transcribed, and they'll pair together. So you really only need a slight amount of pausing for this to occur. Now, if you have no tryptophan whatsoever and you have this ribosome, it's going to sit there at the adjacent tryptophan codons. Well, we're going to get synthesis of those structural genes. We will then be synthesizing tryptophan. Eventually, that ribosome can find a tryptophan-specific tRNA, but we've already controlled transcription of the operon at this point, so it's really a moot point. So again, we can have the ribosome continue to sit on this leader sequence RNA at the tryptophan codons, waiting and waiting for a tryptophan-specific tRNA, or if we have, just fairly low levels of tryptophan and it transiently pauses at the tryptophan codons to wait for a tryptophan tRNA before moving on. Either way, we have slowed down the ribosome enough and allowed transcription of regions 2 and 3 to occur, and they're going to pair to one another. So this eliminates the possibility of having that terminator sequence form, because if 2 and 3 are paired, 3 and 4 will not pair. So it's a little difficult to imagine this in your head because we're having to imagine transcription co-occurring with translation. And then the transcription of an RNA and the rate in which it is translated in region 1, that's going to control whether or not we transcribe our structural genes. And again, we are either only making a very short peptide between the start codon and the tryptophan tRNA or adjacent codons. So either that short little peptide or slightly longer peptide that's going to include the entire region 1 translated. This peptide has no function. It's just going to be degraded by the cell. And it's also important to note that that's the only open reading frame in the entire leader sequence. So the start and stop codon, the only ones in that sequence are located within region 1. Regions 2, 3, and 4 are not ever translated. They're simply transcribed to have a regulatory function on RNA polymerase. Okay. Now that we know we have those structural genes that do need to be translated after transcription, each of the structural genes will have its own Shindell-Garneau sequence, start and stop. And that type of open reading frame is only going to be found within region 1 of this leader sequence, but it will have individual open reading frames for all of the different structural genes. Here's just kind of another look at this. So actually, I didn't mention in the beginning, but regions 1 and 2 actually compare with one another as well. But it's very rare to see this happen. You'll notice that regions 1 and 2, when they pair, we automatically are going to form that terminator sequence because 3 and 4 will pair. But since we know that transcription and translation co-occur in prokaryotes, it's pretty rare for this to occur where we have no ribosome binding of this RNA. And so while regions 1 and 2 can pair, we usually only see 2 and 3 pairing or 3 and 4 pairing. And so in the second part of the slide, we're looking at what's going to happen when we have insufficient levels of tryptophan in the cell. We have enough of a decrease that we have lower levels of tryptophan than specific tRNAs, and our ribosome is then going to pause or take a little bit of extra time to synthesize through those adjacent tryptophan codons. Now we're going to form a loop between 2 and 3. This does not function as a termination signal. We will continue to get our polymerase, our RNA polymerase, to move forward and transcribe the structural genes for tryptophan biosynthesis. Now at the bottom, we see that we have enough tryptophan and therefore enough tryptophan-specific tRNAs in the cell that it's very easy for the ribosome to translate right through those, and then it's going to translate all the way to the stop codon. Once it's at the stop codon, it is physically blocking a region of part 2, and that's going to mean that we get 3 and 4 pairing, and then because we have a string of U's at the end of 3 and 4, we are going to stop transcription. So we want to do a couple of review questions about the tryptophan operon. So if you take a look at this leader sequence, I want you to think about what part of the leader sequence actually gets translated. Is it all of them, or is it just a portion of those regions of RNA that get translated? So the answer is the only part that gets translated is region 1. So we are either going to make a little bit of a peptide between the start codon and the tryptophan codons, or we can make a slightly longer peptide when going from the start codon all the way to the stop. Again, there's no start, stop, or Shindell-Garneau sequence anywhere else in the leader sequence, so just for region 1. And then each of our structural genes will have their own independent Shindell-Garneau and open reading frames for translation of those specific enzymes. Okay, so which scenario is going to generate the longest leader peptide? And I'm referring to that regulatory peptide that is synthesized by the ribosome. Okay, so this will be under high tryptophan levels. We'll get a slightly longer peptide synthesized at this point. It'll be, again, the region between the start and stop codon of region 1. And then under low tryptophan levels, we would just see a shorter peptide from the start codon up to the tryptophan adjacent codons. And so, again, this is a regulatory peptide. It has no function, and it will just get destroyed by the cell. Okay. I want to go over a couple other thought questions before we move on to eukaryotic transcription regulation. So I want you to look back here and imagine, what if you had additional adjacent tryptophan codons? Let's say instead of 2, maybe you had 3. And think about what that would do in terms of regulation of the transcription of the structural genes. By adding a 3rd codon there, would we actually increase the level of attenuation of transcription, or would we decrease the level of transcription attenuation? Think about that. And so what we would actually expect to see is a decrease in transcription attenuation by adding in additional adjacent tryptophan codons. So by having adjacent ones next to each other, 3 instead of 2, the likelihood of causing a ribosome to pause increases. And we know that ribosome pausing at the tryptophan codons always is going to result in regions 2 and 3 pairing with one another, and then we'll get transcription of those tryptophan structural genes. So in a way, we've made the ribosome a little bit more sensitive to tryptophan levels in the cell. We would actually synthesize the tryptophan biosynthetic enzymes at a higher frequency, the more adjacent tryptophan codons we would have. Now, if we only had one tryptophan codon there, the chances that under low tryptophan conditions that we just happen to have a tryptophan-specific tRNA handy for the ribosome, that actually increases. So we're going to be less sensitive to our tryptophan levels because by chance we could find a tryptophan-specific tRNA and move directly to that stop codon. That's going to result in our terminator loop forming, and then we're going to terminate transcription more when we might actually want to be transcribing those genes because we're running low on tryptophan. So again, increasing the number of codons there is going to increase the chances that the ribosome pauses and that we don't get attenuation of transcription. Then, if we have only one tryptophan codon or none, we're going to lose our sensitivity level to tryptophan in the cell. We're going to be more likely to attenuate under low tryptophan conditions. And so for your exam, you should think about other scenarios that I might ask. So what if we prevented regions 2 and 3 from actually pairing to one another? Let's say we removed part of the complementary sequence of region 2, and then it can no longer pair with region 3. Therefore, we would get transcription termination regardless of our tryptophan levels. So we would always have regions 3 and 4 pairing. Therefore, we would always terminate transcription ahead of those structural genes. Likewise, if we prevented 3 and 4 from binding, or if we mutated part of the poly-U sequence that's just following region 4, that would result in blocking our transcription termination. So we would actually get synthesis of the structural genes even if we didn't need them. So this is a very complex, elegant way that the cells have evolved to actually precisely control the levels of tryptophan biosynthetic enzymes based on how this RNA is folding and how the ribosome moves on that RNA. Okay, we're going to move on and introduce some of the regulatory mechanisms for transcription that are specific to eukaryotes. And so we have to remember that we're talking about the cells that, we have all the same DNA, but it differs how we actually use that DNA. And so we have some genes that can also be expressed in responses to environmental factors. Perhaps they're normally wound up in nucleosomes under healthy, normal conditions, or we can have them unwrapped. And so I want you to think about the context of chromatin when we think about how all of these different regulatory mechanisms can control transcription in eukaryotes. So these are going to be a series of signals that the cell receives, and we will be able to turn on and modulate our gene expression accordingly. And so this is basically our current models that we find in eukaryotic transcription. So that is, we have heterochromatin, which is very dense DNA wrapped around proteins. Those proteins are wrapped around each other, etc. We have to expose DNA, as shown on the screen, to actually get transcription. So that's going to be occurring through a combination of histone acetyltransferases, acetylating lysine residues on histone tails. That's going to allow bromodomain containing proteins to bind. So those are creating a binding site. So these are chromatin remodelers that will have either a bromodomain to enhance transcription at a particular region, or we'll have our chromatin remodelers that have chromodomains, and that's going to favor making heterochromatin more dense. And so by exposing a promoter region and also exposing other distal sites such as enhancers or insulator sequences, we can then have a variety of different transcription factors come into play to enact transcription. And remember, for eukaryotes, we have general transcription factors, so TBP, TF2D, etc., along with the RNA polymerase. But we're going to rely on a lot of other factors, including mediator for most genes in eukaryotes, acting as a co-activator by transmitting signals from distal sites. So we're showing here we have activators interacting with enhancer regions on the DNA that's being bent around to the promoter region. And often what we see is that mediator is going to be connected to both the RNA polymerase and these activators, transmitting a signal to increase transcription. We can also have repressors that actually block the ability of activators from interacting with mediator and RNA polymerase. So there's a series of different possible ways that our cells are going to regulate transcription at all of the different genes. And so transcription factors, to remind you, typically have two main domains. They have a portion of the protein that binds DNA, and then they have a portion of the protein that interacts with another protein, basically gives it a specific activity. So you can have a series of transcription factors that all bind the same site on the DNA, but let's say one of them has an activation domain that functions as an activator of transcription. Another one, their active part of their domain, could be to repress transcription. So you can localize a repressor and an activator to the same site in the DNA if they have the same binding domain or DNA binding domain, but their activity can be different. And so you can control, instead of controlling where they bind, if they're binding the same site and competing for the same site, you would then be able to control transcription through just the enzymatic levels of different transcription factors. So we will later on talk about how these transcription can be regulated in a concentration-dependent manner with regards to its different transcription factors. Here on the screen, I'm just showing you some examples of typical DNA binding domains and activation domains of transcription factors. However, I'm not going to ask you to recall any of these particular names. And so it turns out that most transcription factors actually function as dimers. So remember, this is copies of proteins that they work together to have a quaternary structure for a particular function. Now, if you're looking on the screen here, I'm showing you that we actually have three factors, so A, B, and C, that are transcription factors. They all function as dimers, and they function by dimerizing with each other. And so based on the combination of transcription factors in that dimer, through the expression of three factors, we can actually see transcription factors that recognize 6 total binding sites. And so notice that they are made up of what's called the inverted repeats. So in the lower part of the screen, these are complementary sequences that are going to bind DNA. And so we see our transcription factors functioning as dimers by binding the DNA while they're still associated to one another. And so depending on the actual sequence that they're binding, you can have 2 copies of transcription factor A binding a particular site. What if transcription factor A dimerizes with B? Well, that allows it to bind an entirely separate regulatory site. So these are called both homodimers and heterodimers, depending on if it's 2 copies of the same factor binding itself, or if we're having 2 different transcription factors come together. So again, this is going to allow the expression of 3 different proteins to then act on DNA at 6 different sites. So we've really created a diverse way in which these proteins can regulate transcription. And again, it's through different dimerization, homodimers and heterodimers. And so when we have to think about the fact that transcription regulation in eukaryotes is going to involve chromatin remodeling as well as the combined efforts of different activators, repressors, co-repressors, effector molecules, etc. So what we will tend to call this is combinatorial control of gene expression. So multiple transcription factors, multiple DNA sequences are going to play a role. and it's much more complex than what we see in our prokaryotes. And so the first example of combinatorial control that I'm going to discuss is called the yeast gal genes. And so these are no longer going to be referred to as operons. That's a term that is reserved to describe prokaryotic transcription regulation. Remember that every coding region in a eukaryote is going to have its own promoter. We can alternatively splice that transcript that is made. However, we are only going to get one single protein per transcript. So these are monocistronic RNAs, whereas in prokaryotes we see a single promoter regulating an entire set of genes. And so those are operons, and this is just a general promoter region that we're going to be talking about in eukaryotes. And so before we discuss the GAL1 gene regulation, we have to introduce a couple specific chromatin remodelers. So again, recall that histone acetyltransferases, or HATs, are going to acetylate lysine on histone tails, recruiting bromodomain containing proteins. There's one bromodomain containing protein that I'm going to have you learn in this class, and that's a complex called SWI/SNF. And so SWI/SNF is actually going to further unwind the DNA and actually really expose a promoter region for transcription to take place. If you notice at the top of the screen, there's a promoter region that's actually wrapped around the nucleosome. We can loosen that interaction through HAT activity, but we know that that's not sufficient in most cases to actually expose a promoter region. We need to physically remove that part of the DNA from the nucleosome. And so we see that there are a series of additional factors that can come into play here, transcriptional activators interacting with SWI/SNF, along with SWI/SNF binding the acetylated lysines of our histone tails. There's a combination of factors that's basically going to remove the promoter region from the nucleosome and then allow the general transcription machinery such as TF2D, TBP, RNA polymerase, it's going to make room to recruit them to our polymerase here. And so it's going to be, again, combinatorial control. First, we are acetylating lysines on histone tails to loosen up the DNA. That action alone is going to recruit a bromodomain-containing protein called SWI/SNF. It's a very kind of general bromodomain-containing protein that works to modify the chromatin, removing the promoter region from the nucleosome. And again, lastly, we can then recruit all of our general transcription machinery. So we have a few steps that we have to get to before we can even recruit RNA polymerase. And then once we've recruited RNA polymerase, we can actually modulate its activity further through addition of other complexes such as mediator, activators, repressors, etc. And so again, we're going to cover a few different examples of things that are really considered unique to eukaryotes. We haven't seen much of this in prokaryotic gene regulation for some reason, because we have we're not making heterochromatin in prokaryotes, for example. So we're going to go over how galactose metabolism is controlled in response to the galactose sugar. We're going to talk about something called yeast mating types. We're going to go over hormone signaling. We're going to talk a little bit more in depth about how insulator sequences function. Then we will discuss an example of genetic imprinting, meaning epigenetic changes of your DNA from your parents on your inherited chromosomes are actually going to be able to control gene expression. Then we'll talk briefly about how 5' and 3' UTR regions influence transcription in eukaryotes, and then how RNA can interfere with transcription in eukaryotes, and then lastly, how transcription factor gradients or concentrations can influence transcription. So these are a lot of different examples, so I want you to be able to, when you're studying for your exam, kind of check off when you understand each of these individual examples. So first, for the yeast GAL1 promoter, this is going to control genes that is a gene that's responsible for the uptake and metabolism of galactose. So again, this is a gene that you're not going to want to transcribe unless you have galactose present. So some sort of signal needs to reach the transcription machinery at this particular promoter to repress it under certain conditions where you don't have any galactose, and then to activate transcription of GAL1 when we do have galactose present. So notice here we have the galactose GAL1 structural gene next to the transcription start site in yellow. Then upstream we have a collection of sites that are actually called GAL4P binding sites. These are, again, just upstream activating sequences or kind of upstream enhancer type sequences that are going to help control the rate of transcription at this particular promoter. In yeast, we often will call the proteins by adding a P to the end of their name. So this is the GAL4 protein, and so I will either refer to it as GAL4 or GAL4P, as well as the other proteins in this pathway. They both mean the exact same thing. It's just the nomenclature that we use for yeast genes. So GAL4P has several different domains. It has the normal domains that we would expect for transcription factors. One would be a DNA binding domain, where it interacts with the DNA, an activation domain, where it interacts with other factors, and we have a dimerization domain, which again is going to be very common in transcription factors because many of them only function as a dimer. So in this case, GAL4 functions as a homodimer. It needs 2 copies of itself, and when they are interacting together in a quaternary structure, they combine the DNA. And now we have this free activation domain. So we have a repressor, which is GAL80, which is going to interact with the activation domain of GAL4. So this is basically our default situation where we have GAL4P synthesized, dimerizes, binds at this upstream sequence. Then we have GAL80, which is always, again, expressed and then bound to GAL4. So it's preventing GAL4's action. This would be an example of a co-repressor. So GAL80, by blocking the activation domain of GAL4, that prevents it from transmitting a transcriptional activation signal to our RNA polymerase. So regardless of whether or not we have our general transcription machinery associated here at the promoter, we are also relying to get transcription of GAL1. We're relying on other signals that come from the cell. And so the last factor in this pathway that we'll discuss is something called GAL3. So GAL3, we consider it, we give it the name transducer because it is going to actually cross the nuclear envelope and interact with the DNA based on whether or not it is bound to galactose. So we have galactose. If it's present in the cell in any appreciable amount, it's going to bind GAL3. Binding of galactose to GAL3 causes GAL3 to undergo a conformational change, which allows it to be translocated from the cytoplasm to the nucleus. So normally, GAL3 is just found in the cytoplasm, and in this situation, we've translocated it into the nucleus so it can function as a transcription factor and modulate the activity of GAL4. So when you have GAL3, 4, and 80 all bound together, like on the bottom part of that figure, we actually have kind of uncovered the activation domain of GAL4. So by having all 3 bound together, the activation domain of GAL4 is free to interact with the RNA polymerase to stimulate transcription. So we're needing to remove nucleosomes at the promoter region through a series of chromatin remodelers. Once we've done that, we're still waiting for other signals within the cell to activate transcription. Again, we call that a type of combinatorial control of transcription. So the way that we actually are going to modify our chromatin is through, this is the specific hat that is going to acetylate the nucleosomes adjacent to the GAL1 gene promoter. So this is called SAGA. So SAGA is the histone acetyltransferase. Sometimes we call histone acetyltransferases co-activators because they can actually modulate transcription without actually physically touching the DNA. So notice here we have Gal4 and Gal80. They're bound at that upstream activation sequence. Then we have part of our promoter region exposed, and we have the rest of the gene sequence actually wrapped around in nucleosomes. So when you have all three factors together, you have galactose present that's caused a translocation of Gal3 into the nucleus. We have Gal3, 4, and 80 all bound together. Well, it turns out that that is a signal that recruits SAGA, the histone acetyltransferase. And so we haven't really talked about in this class how the HATs and HDACs get recruited to histone tails to function. So this is one example. So you have some transcription factors in a particular orientation and conformation, and that will serve as a recruitment for this HAT called SAGA. So SAGA is then going to begin acetylating the lysines on the histone tails adjacent to the Gal1 promoter. And it also is going to help recruit TATA binding protein to help bind that TATA box. So we're loosening the DNA interaction between DNA and the histones. We have kind of revealed a little bit more of the promoter region and begun recruiting our general transcription factors, and that is through this effect of SAGA. Now, SAGA, we know, again, is a histone acetyltransferase, and by acetylating lysines, it recruits chromatin remodelers. And again, a specific example of a chromatin remodeler here could be SWE/SNF. And so any bromodomain containing chromatin remodeler will be again recruited to that site to further unwind the DNA, remove the entire sequence that needs to be transcribed from our nucleosomes. Finally, through the action of all of these different proteins, it's highly coordinated, we can achieve transcription of the Gal1 gene. So again, we have to have certain signals that are transmitted through the presence of galactose in the cell. That causes translocation of Gal3 into the nucleus. It binds to Gal4 and 80, that complex. Then it shifts that complex conformational change around, exposing the activation domain of Gal4, recruiting a histone acetyltransferase called SAGA. That's going to further modify our chromatin, making it more like euchromatin, and it's going to recruit chromatin remodelers, in this case, SWE/SNF, through a bromodomain. So all of this coordinated efforts from all of these different proteins and signals is required to achieve even one transcription, round of Gal1. So in our eukaryotic cells, we are generally not just turning genes on and off and basal. We can regulate transcription at many different levels, anywhere between 0% of possible transcription and 100% of possible transcription. And that's through the fact that we have a number of different events and proteins that are involved in achieving just one round of transcription. So you can imagine all of the different types of scenarios that could occur between combinations of signals coming within the cells, acting as allosteric regulators of different transcription factors, and different combination of transcription factors are going to recruit either HDACs or HATs that will recruit other chromatin remodelers, etc. So eukaryotic gene regulation is much more complex than what we see in our prokaryotic cells. And so for this clicker question, I want to ask you, what would happen if you had a mutant Gal4? So just kind of assume that, Gal4's function is disrupted. Okay, so in this case, Gal1 would never be expressed. So if we have a non-functional Gal4, let's say maybe its DNA binding domain is still functional, but its activation domain is not functional. Well, we're not going to get ever the recruitment of all three transcription factors required for Gal1 synthesis, because we need both Gal4, Gal80, and Gal3 to come together to recruit histone acetyltransferases, other chromatin remodelers, and to directly interact with our general transcription factors. So imagine either if it didn't bind the DNA in the first place, we would not get recruitment of any of those other transcription factors required for Gal1 expression. If it was the activation domain that was mutated, we could get recruitment of some of those factors. But again, we're missing some of the pieces of the puzzle here. So it's important to think about what would happen under different mutation conditions. Okay, I'm going to go over an example about something called yeast mating types. And I want to preface this by saying I'm not going to ask you about the specific genes discussed in this example. It's just an interesting and kind of elegant way that yeast control gene expression through different combinations of transcription factors. So again, we're just going over this as an illustrative example, and it's not something that I'm going to specifically ask you on your exam, and therefore the genes that we're talking about here are not going to be listed in your study guide. And so when we have our yeast cells, they exist as both haploid and diploid cells. And so we actually will use an A cell and an alpha cell. Those are two different haploid cells that will express A-specific genes if you're an A cell, and alpha-specific genes if you're an alpha cell. So when those two fuse, they become an A/alpha diploid cell. Then we're going to want an entirely different type of transcription regulation when they're together. So we have, again, specific genes for haploid A cells, haploid alpha cells, and we're not going to need those genes anymore once we become a diploid cell. So there's an interesting way that our A and alpha cell transcription factors combine to control gene expression, and that is going to be particular for whatever form they're in, haploid or diploid. And so there's a specific sequence called a MAT-A or a MAT-alpha transcription sequence. And so this is going to help control A-specific genes and alpha-specific genes. And so our haploid cells only have one or the other. And then in our diploid cells, we are going to have both of these regions. Again, this is a regulatory region where transcription factors will bind to control expression of either A-specific genes or alpha-specific genes. Okay. And so this is what some of these scenarios are actually going to look like. And so if we starting with part A, we're looking at an A cell, which is haploid. So at that MAT locus, the MAT-A locus in their genome, that is going to cause expression of a factor called alpha-1 and MCM1. Now in B, for our alpha-specific cell, which is haploid, that MAT locus is going to control expression of a couple other different factors, alpha-1 and alpha-2. Both the haploid cells and the diploid cell will express a separate transcription factor called MCM1. And so what we're going to see is that the combination of MCM1, which is universal, with transcription factors that are specific for the different haploid cells are going to control gene expression in different ways. So for our A-specific cell, MCM1 goes and sits on the promoter region of where we are able to control A-specific genes. So MCM1 binds that promoter region. We've turned on A-specific genes. Now our alpha-specific genes will require an additional transcription factor to be turned on, which the A cells actually lack. In our haploid-specific genes, just another set of genes specific for haploid cells, doesn't require any additional transcription factors, but as we'll see in a diploid cell where we don't want those haploid-specific genes, we will actually be able to turn it off with a different combination of transcription factors. So in B, this haploid cell is going to use MCM1 and 2 copies of alpha-2. So this is a heterotrimer that binds a slightly different sequence within the promoter region of A-specific genes. And in this case, it actually turns expression off. So this is to reiterate that different combinations of transcription factors combine different sequences within the promoter region or adjacent to the promoter region, and they can either function to turn transcription on or off. Now, in the alpha cell, we want to turn on those alpha-specific genes. So the A cells actually don't have the combination of transcription factors required for that, but of course the alpha cells do. And so the combination of the factor alpha-1 with MCM1 is going to bind that promoter region of the alpha-specific genes and turn them on. And again, these haploid-specific genes are universally expressed in the haploid cells. We don't need to regulate them in any way in the A cell or the alpha cell. Now, once we have an A/alpha cell, a diploid cell, we actually lose expression of alpha-1. So that gets blocked in a diploid cell. So the remaining factors that are expressed that we've talked about are A1 and alpha-2. So, and we're still going to express MCM1. So if we want to turn off our A-specific genes, turn off our alpha-specific genes, and turn off our haploid-specific genes, we can use different combinations of the transcription factors present in diploid cells to achieve this. So our A-specific genes are going to be turned off the same way that they are in the alpha haploid cells, because we can still express our alpha-2 protein, and that heterotrimer is still going to be expressed in the diploid cells. So it sits on the promoter region, turns off transcription of the A genes, the A-specific genes. Next, we can use the same mechanism that the A haploid cells use to turn off transcription in their cells. Same way in the diploid cells. We don't require any additional factors to turn it off. It's the opposite. We need other factors to turn it on. And since we're not expressing alpha-1 in our diploid cells, we can't have that combination of alpha-1 and MCM1 like we do in our alpha haploid cells. So that remains off in our diploid cells. Lastly, we need to be able to turn off these haploid-specific genes because they're only something that haploid cells need. So to do so, we're going to take a heterodimer of A1 and alpha-2. That combination of transcription factors is going to bind the promoter region of the haploid-specific genes and then turn expression off there. So notice that each cell has the exact combination of transcription factors it needs to turn on and off the different types of transcription factors or genes that are required for that type of cell. So each type of cell is going to have different requirements of what it needs to function, and we will use these different combinations of transcription factors to bind sequences to modulate activity. And by combining the two haploid cells, we're bringing together two transcription factors that would never occur together in a haploid cell, but in a diploid cell, they come together and they can function to turn off some of these haploid-specific genes. Okay, so we're going to stop there and then pick up on some of the other examples of eukaryotic gene regulation on Friday.